the section on microbiology, for the body as host. And I'm Dr. Sam-Yellowe from Cleveland State. And so I've done this, about 3-4 years by now. And so, we're here again to do this. And so in this section of things, the idea is to review in some respects and others, get some new information about microbiology. And so we'll just begin since we've already lost some time. So for today, the notes that you have, we have some key concepts that we want to get across. And so we have the first item, the human microbiome. We want to understand just truly how diverse the human microbiome is. And within the body, even though we're going to spend some time looking at the oral environment, we still need to remember that the human microbiome essentially covers, the whole body. And so we have, different niches, within the body where we find organisms, and those organisms vary. And so part of what we're going to be trying to do is get a general sense of what these organisms are, and in particular, focus on the ones that we find in the oral cavity. And, in a few places, we will mention significant organisms that are normally GI tract organisms, but show up in cultures that are obtained from the oral cavity. And one such organism is Helicobacter pylori, and when we get to that point, we will say some more about it. One of the other concepts we want to get across today is the fact that prokaryotes and eukaryotes have distinct characteristics. So we want to kind of remind ourselves, what prokaryotes are, what eukaryotes are. And among the prokaryotes, we want to remember what the gram-positive organisms look like, what the gram-negative organisms look like. And the ones that are non-gram-staining, acid-fast organisms, remember what those are and why they're acid-fast and so on. And so that kind of ties in with the third concept there, understanding why gram-staining is important, and how that, along with serology and biochemistry and genetics and typing for organisms and strains, why that's important. So the fourth item on here, we want to also kind of get an understanding, how do we study these things? So, for a good 50% or so, 60% of the organisms of the bacteria, we don't have them growing in culture. In other words, we cannot culture them. We don't know what conditions they require. We don't know what they need for nutrients. And so, many of these have been identified using molecular methods. So in other words, the DNA, from communities of organisms, biofilms have been obtained, and that DNA, has been amplified, and the information, from the sequences of these amplicons have been blasted, to databases, and from that blasting, we have matches to different phyla of bacteria, let's say, and then the ones that did not match to any, we have them in essentially new phyla. And this pretty much awaiting, a better understanding of how to grow them and describe them methodologically and so on. And so bioinformatics, is a very important area, and the things that we'll talk about, you see us, mention that as we go on. And then so we want to know the distribution of the normal organisms, the normal microbiota that we have in the body. So what's normal, what's commensal, what's potentially pathogenic, meaning they opportunistic, and what are the true pathogens. Okay, and so we want to be able to do that. And so we know that bacteria colonize different parts of the body. We already mentioned the GI tract, oral cavity, the urogenital tract, and we have biofilms that form in these areas. And so we need to know, what this distribution is. And then finally, on the concepts we want to kind of get across today, we want to become familiar with the major phyla of bacteria that we have in the body. So, we have several different phyla of bacteria. Not all of them are found colonizing the body. but we have ones, you know, that we associate, you know, very prominently with the human microbiome. And so we're going to need to know them. We're going to need to know examples of organisms found, you know, in those phyla, and specifically when we get to the point where we're talking about the organisms in the oral cavity, commensals, as well as opportunistic organisms, as well as the pathogens, we want to be able to identify them within these phyla. And so we will be talking about that. So of the four major ones we have here, the actinobacteria, Firmicutes, we have a proteobacteria, which is a really large phylum of gram-negative organisms. We have the Bacteroides as well, and so we have an important uncultivable phylum that was described recently, again using the bioinformatics methods that I just mentioned to you, and that's the TM7. So we will, you know, say a few things about it, you know, when we get to that point. So, in order to get us beginning to look at the organisms that are important for disease, right? We want to also make sure that we remember what folks' postulates are, and you know, as much as you know, the postulates don't satisfy every single pathogenic state, you know, where we have organisms causing infection, and that means bacteria, fungi, parasites, and so on, right? There are still things to take away from these postulates. And in the places where we have difficulty fulfilling every step of those postulates, we will explain why. And so that way we'll have an appreciation for that. So we have the organization of organisms, right? So we have the, you know, the domains, which, you know, I figure we're all familiar with. So we have the eukarya, here we have the archaea and the bacteria. And so the eukarya comprise all the eukaryotes, okay? So that means, you know, the large group of organisms. So out of the 5, you know, kingdoms, 4 of them are eukaryotes. And so in the one kingdom of the bacteria, or the eukaryotes, we have the archaea and the bacteria in that group. Okay, so again, you know, depending on what you're reading, you know, you have discussions going back and forth that the 5 kingdoms, you know, that we currently have, you know, are too small, they're too narrow, and you have calls for expanding those kingdoms, right? But as far as, you know, for the purposes we're working with now, we'll stick with the 5, okay? So if you, you know, if you're interested in that area and you know, pick up something to read and you're trying to, you know, understand why we're still sticking with 5, you know, you'll see that there's a lot of discussion going on with regards to, you know, the narrowness of the 5 kingdoms, and you know, we need to expand them. So we're going to stick with the 5. And so of the 5, 4 are eukaryotes. Okay, so in animals, plants, protists, you know, fungi, those 4 are eukaryotes. And then 1, okay, the 5th one, is prokaryote. And so we have, amongst both groups, you know, eukaryotes and prokaryotes, we have, you know, a lot of diversity, and we have, you know, complex ecosystems that define, you know, each of the groups. And within the groups, we have, you know, symbiotic organisms. So we have, you know, symbiotic relationships that are not harmful to either host. So we have mutualism, we have commensalism that will kind of fall into those categories. And amongst the commensals, we can have problems if, you know, some of those commensals become opportunistic, right? And then we have, you know, parasitic situations. So this is when we have organisms that are obligately dependent on a host, and they will parasitize that host. So for what you want to remember, again, within this symbiotic group, we have these, you know, there are other subgroups, you know, underneath mutualism and commensalism, but we're not really interested in those, you know, right at this point. But we want to remember the major groups. And so we can describe the organisms as being non-pathogenic, okay, you know, within these relationships. And as well, we can describe them as being pathogenic if they're disease causing, okay? So amongst the non-pathogenic, just make a note that we can have opportunistic organisms there. In other words, under normal circumstances, they will not be pathogenic. They will not cause infection. But depending on what's going on with the individual, if you have chronic conditions, chronic illnesses, these organisms can now take hold, and in this case, they behave in opportunistic fashion. Okay, did that concept make sense? Okay. All right, so I always like to include this here, just so that we can kind of get a sense of the diversity of the microbiome, and there are really just a couple of things I want you to take away from this big diagram. And that is, if you look around the edges, okay, of this, these circles over here, you see the different examples of organisms, representing the different phyla, the 4 of which we already mentioned. And as much as we have, all this large, and again, this figure doesn't include every single thing that can be included, but as much as we have that, okay, in the human microbiome, as we've already said, right, we have, the 4 major ones, okay, that we've talked about. So we all need to remember them. The Actinobacteria, we need to remember Bacteroides, Firmicutes, and the Proteobacter, okay, or Proteobacteria. So we need to remember these 4. And we need to remember them in terms of, their gram staining, which we'll get to in a minute. We also need to be able to identify examples that fall within the 4 phyla. And so the one that's the most diverse in here is the Proteobacteria. So we have 5 subgroups underneath it, right? So we have, the alpha Proteobacteria, we have the beta Proteobacteria, we have epsilon, we have delta, we have gamma. And so we're going to see representations, of these in here. And this is a huge gram-negative group of organisms. The second thing that I want you to kind of take away from this diagram, this figure over here, is the fact that when you look at it, and you probably can't see very much from just looking at the screen, but hopefully you'll take a look at it on your computer. And what you want to appreciate is the number of those organisms that are commensals versus those ones that are pathogenic. So the commensals have the circles, and the pathogenic ones have the stars, or they're starred. And so the point to make with this is that we have way more commensal non-disease causing organisms than we do the pathogenic ones. And this is true not only in one location in the body. So this is true if we look at the oral cavity. It's true if we look at the skin. It's true if we look at the GI tract, the urogenital tract, respiratory tract, right? We have more commensal organisms present than those that are frank pathogens, okay? And, we have a few amongst, the commensal organisms that can become opportunistic. Okay, does that make sense? This is going to be important when we look at what's going on with, inflammation in the mouth, around the gingiva and areas like this. Looking at the organisms, we're going to find out that this same thing holds when we get there. So I included this figure just, for really just a main point. Okay, kind of based on what we already saw in the previous slide. And in this paper by Blum, he goes through again looking at the different phyla, the organisms within those phyla, and their distribution amongst healthy individuals and patients of different kinds. So we'll say more about this later on, but again, the whole point of this is when you look at this figure, again, you can see, the large distribution of, things like Firmicutes, okay, in different areas in which we have a large number of organisms, within that group. And you see the differences in the profile between healthy individuals and individuals, in various disease states. And this particular figure just lists, a few examples of something like, inflammatory bowel disease, type 2 diabetes, and necrotizing enterocolitis, and just showing the shift in the distribution of groups of organisms, represented by the phyla. So, again, we're seeing in healthy controls, a lot of, Firmicutes organisms, and you see the shifts in the 3 disease states, that group of organisms is altered. And so these days, and this has been going on now for about the last 10 years, and I'm sure you've already talked about it in your other classes, or you're aware of it and so on. There's a lot of interest in trying to understand in a better way what the normal commensal organisms do for the body, right? What is it that they provide, okay, in a healthy state? And what then happens when populations are altered? And so this is being looked at in virtually everything. So it's being looked at, for the health of the mouth, for the health of the GI tract, in a variety of, chronic diseases, all the way to neurological, conditions, including things like Alzheimer's. There's a big interest, in trying to understand the contribution that the bacteria make, to healthy states. And I don't think it's escaped your attention, to see people talking about probiotics and prebiotics virtually everywhere you look. Okay, the last 10 years, it's kind of just gone up, because of the interest in these organisms. And so, and these days it's not unusual to hear or read about, fecal transplantations. Has anybody not heard about that already? Fecal transplantations, right? And again, the idea is if you have a dysbiosis, right, can you restore the health of the population by introducing new good bacteria from a healthy individual, okay? And so it's a big area of research, of medicine and so on to understand these things. And then, so of the study type things, I included this one over here just to have us get an appreciation for the way in which these organisms are studied. So remember we said, about 50%, 60% of organisms we cannot grow, right? But we know they're present because we can identify their DNA. And so if we can identify their DNA by doing polymerase chain reaction, for example, using a 16S, subunit RNA, and if we can identify the DNA, we can sequence that DNA, we can blast the DNA into the databases, we can get information regarding, what phylum, they're matching into, or what they belong. And so that's not the only thing that can be done. And so what this figure is showing you is that the data that's obtained can be analyzed in different ways. And so pretty much, if you look at each of the bubbles, it shows you, right, you can gather information, you can do transcriptomics, genomics, you can do metabolomics, you can find out information just based on that DNA you identified. And, you can begin to do searches to find out, the phyla they belong to, particular, organisms, that they match with and so on. You can look at the SNP profiles on these things, you can do RNA-seq type information, and you can gather all of that and generate profiles of the communities that you have just examined. And so by doing this, we know a lot about, the organisms that we cannot even culture. But based on the profiles, we can begin to use that information to try to understand the type of communities we're finding these cells growing in, to try to understand better how to approach things like prevention and so on. So it's a very hot area of research, and some of the papers that I will mention as we go on are being done by dental researchers, and so it will kind of give you some insight as to some of the things, the possibilities, the potential that is out there to get information that can actually better help in the clinic. All right, so the way that the schedule is organized is that we're supposed to be looking at the anatomy of bacteria, and that's what we're going to be doing, and we're going to kind of spread that out into today and tomorrow. And so some of the stuff is going to be things that you've already, you already know, you've already encountered before. And so it will be a review, and for some it will be the first time, that we're kind of hearing it, and so we're kind of start at the same place. And so what we have up here that we're looking at, we have a prokaryote, eukaryote, so there's some key things we want to get. So the differences, between eukaryotes and prokaryotes is not based on the size of the cells. So, the differences between the two, right, is based on something more fundamental. And I think we all know what that difference is. What's the difference between prokaryote and eukaryote? I heard it. Nucleus, okay? So we have the DNA enclosed in eukaryotes. We have a true nucleus. We have an endomembranous system, right, with the organelles. Right, so we have, the endoplasmic reticulum, smooth and rough, we have the Golgi, and we have a variety of other organelles, lysosomes, peroxisomes, hydrogenosomes, depending on, on what organism you're looking at. And so we have those, basic fundamental differences. In prokaryotes, typically, and I say typically because, we have some very large bacteria that when they were first described were thought to be eukaryotes. But it ended up being that they're actually bacteria, they're just very large. So bacteria have their DNA lying free in the cytoplasm. We don't have an endomembrane system, okay, typically. And so we have, no organelles, right? And so the ribosomes that we have would really be the equivalent, okay, of what you would refer to as an organelle as such. And so we have those distinct differences. Now, both eukaryotes and prokaryotes can have flagella, right? So that's not a distinguishing feature. Does that make sense? Flagella, it can be found in eukaryotes, can be found in prokaryotes. Prokaryotes and eukaryotes can have cilia. So cilia is not distinguishing. Is everybody with me? Okay, so we can't use that to distinguish because we find cilia in both groups. And so other things, that are used to distinguish, right, the ribosomal content. So we have, the 70S, ribosomes that tell us we're dealing with prokaryotes, and then the 80S that tells us we're dealing with eukaryotes. And so whenever these communities, like I just mentioned to you, are examined, right, and, can't culture the cells, but we can find the DNA. So we're using the conserved regions of the 16S, RNA subunit, okay, to do the amplification, okay. And so other things that you do see in here, right? So we have a cell wall in the prokaryotes, but that by itself cannot be a distinguishing feature because we have, eukaryotes, right, that have a cell wall. So plant cells, etc. We have cell walls there. So we can't use them, okay? So we just need to just acknowledge that, yes, it's there, okay, and the type. So even though we have it in both, eukaryotes and the prokaryotes, the cell wall in bacteria is heavily peptidoglycan based, okay? So that's different from what we find in plant cells. And then, of course, as you look through, within, the eukaryotic cell, we have mitochondria, etc. We have chloroplasts, that kind of a thing. And so the endosymbiotic theory, lets us know that the mitochondria and the chloroplasts, derived, from the bacteria, okay? And so we have, the photosynthetic situation, okay, of aerobic, bacteria that, we now have the mitochondria from. And so we need to remember why, and this is because, when we have aerobic respiration, right, and we have, the whole process, of having ATP generated is very similar to what happens on the aerobic bacteria that we have in the membrane, okay? So we need to remember that aspect of things. And so when we look at, what else is found, in all of these cell types have a plasma membrane. So there's a limited membrane on both, prokaryotic and eukaryotic cells. So we have, amongst the bacteria, diverse shapes. And so, we're seeing, the different types over here, and there are others that we could include in here. So we have everything from truly rod-shaped, cocci, and the coccobacillary types. They're not fully round, they're not fully, rods. We have fusiform types, we have, spirokicks, etc., vibrio, cover-shaped looking organisms, and so on. And so what we're not seeing here, which, we'll mention sort of in passing, in one, in one lecture, and then a little bit more detail, towards the end. Organisms, that have, essentially prosthesis. They have the prosticate bacteria, they have appendages on them. And as well, we have ones that are predators, right, that will prey on other bacteria. And so if we were to put them in here, with these shapes, we would have to indicate them with the appendages they have on them. Some of them, actually, bud as if they're, fungi and so on. So we have a lot more diversity than what we're seeing over here, okay? And so morphology is important for distinguishing the cells, the way in which, the cells arrange themselves when they divide, right? When we culture the cells, things that we typically look for, right? When we have the colonies form, we look for pigmentation, the shape of the colonies, if they're smooth or flat or raised. I mean, there's all those different characteristics that we use to describe the colonies. And, and as well, the smell of the culture, the various metabolites we produce, will distinguish the cell types, the different types. And so, as we've already said, we have the group, that we can't grow, but we can use molecular techniques to identify them. And so pretty much kind of a weighting, the time point at which the culture conditions that are required will be now known. And then, we can actually see what they look like, after they've been grown. And these two quotations over here, interesting, for the things that we're going to be looking at. So, the first one, again, has to do with just the sheer number of bacteria that we have in the body. So we pretty much have 10 times more than our normal cells, okay, of bacteria growing. And so, the quotation over here, the question of what it means to be human is more often encountered in metaphysics than in bioinformatics, but it is surprisingly relevant when studying the human microbiome. Right? So we have this sheer volume. And so if we looked at the cells, just the volume of cells, and if we looked at the DNA represented by the cells, right? That DNA, just the amount of it, the different types of it, right, surpasses what we have on our own somatic cells. Does that make sense? Okay? And so, this is, a very significant thing, okay, when we have to think about these organisms that we have, living in various areas of the body and what they're doing. Now, the second quotation there, the term microbiome was coined by Jeffrey Lederberg. And, so when he basically, saw and could appreciate just, how diverse these organisms are that inhabit, our body spaces, right? So he coined that term to be ecological community of commensal, symbiotic, and pathogenic microorganisms sharing our body space, okay? So one of the other terms that you will hear, which essentially means the same thing, these days they use it interchangeably, is the microbiota, okay? So I think most people have heard that term before, and it's pretty much referring to the same thing. So, we've already said a few of the things. So, 10 times the number of human cells in the organism. And so it's essentially an organ unto itself because of the, the different cell types. And, the diversity of the genes, the number of those genes, the term that's used to describe them is the metagenome. The metagenome, okay? And so we have, within the skin, GI, oral cavity, urogenital tract, respiratory, etc. And so one of the things that we're going to want to appreciate is biofilm formation, right? How does the biofilm form, wherever they form, and how does it get established? And so one of the things to mention just now is that we usually start with founder species, and then we build on those with new organisms showing up. And then we essentially have permanent members of the community, and we have transient members. And we can typically find those transient members at the peripheries of the biofilm. And so within the biofilm, right, and as well, in other locations you find the bacteria. So if the bacteria are present, then that means they have their viruses with them, their phage, and as well they have plasmids, right, that can go back and forth. So then we have that. Go ahead.
The second bullet point says 10 times the number of human cells, so the microbiome is considered an organ?
That's the way it's being thought of, because of how large it is, right? And so, and keep in mind that, again, this is the reason why, it's being referred to as that. The microbiome interacts with our endocrine system, with our immune system. It also interacts with our nervous system in very major ways. And so, the whole idea of referring it to as an organ is that it consists of all these different cell types, right, that have these diverse functions, even under normal commensal, circumstances. Does that make sense? Okay. And so, again, we can have a lot of influence on host health, and as we've already said, different locations, and we have patterns, okay, of clustering of these organisms in different places. And so, one of the things, and I'll see if this will work, if not, I'll just say it, maybe it might be easy just to say it. So the way you want to think about the bacteria, right? So if we have prokaryotes, and we're all familiar with dichotomous keys, right? So if we have prokaryotes, we have, the ones that have the peptidoglycan cell wall, okay, which would be the regular bacteria. We have the ones that have the pseudomurine, the archaea, okay? So in some respects, the archaea, more eukaryote-like than they are prokaryote. That's we'll get to that. And so you have these two big groups, right? So the archaea, okay, we have, separated. And the ones with the peptidoglycan, right, constitute the major group of the bacteria, right? And so you have, a couple of groups that are cell wall-less, okay? So we'll talk about mycoplasma and so on. And then, so amongst the bacteria, when we do the gram stain, we can separate them into gram-positive, gram-negative. Is everybody with me? Right, so gram-positive, gram-negative. So we have organisms that are poor gram stainers, okay? So mycobacteria, for example, gram variable, okay? So we use the acid-fast, acid-fast staining, and so the acid-fast positive, and so we can, tell that grouping there. And then so amongst, the gram-positive organisms, we have those that have high GC content. And then we have those that have low GC content. So the nucleotide bases, okay, your guanine cytosine. Some have very high content, almost as much as 70%, and then others very low, okay, among the gram-positive organisms. And then amongst the gram-negative organisms, right, we have this huge group, which will encounter many of the ones we find in the mouth, okay, all in this category, right? So we have, gram-positive, gram-negative organisms. We'll see a lot of these gram-negative ones, in the oral cavity. And so we have those that fall into the proteobacteria group. Remember the four phyla, right? And then we have non-proteobacteria group. Amongst the proteobacteria, we have alpha proteobacteria, beta proteobacteria, delta, epsilon, and gamma. Okay? Did everybody kind of get that? Right, so we have, alpha, beta, we have delta, we have epsilon, we have gamma, proteobacteria, okay? And so within this, large group, we will find a lot of the, pathogenic organisms, okay, that we're going to be talking about, and so on. So as we get to the point where we're going to be talking about organisms, we want to be able to kind of fit them into these phyla. So again, of the pathogenic organisms, right, so we have, the group of organisms that we can find, the different areas, bacteria, archaea, we have fungi represented, we have viruses, bacteriophage, so on, multibacteria, and then we have, protozoans. And so, although protozoans are not things that people typically think about, in the oral cavity, right? So when you're thinking about organisms in the mouth, you're not thinking about protists, but we do have a couple of important ones that we're going to mention. Okay, so we have, trichomonas, that's in there. We have entamoeba, that are pretty important depending on where, you live, and so on. Those, might be important organisms that are in the mouth. The trichomonas tenax and entamoeba gingivalis. These are protozoan organisms, and they're important in the oral cavity. And so with the bacterial size, so part of looking at the anatomy of the bacteria is trying to understand not just how they're made up, but differences in the size. So we can have as small as 1-2 microns to as large as 750. And so the typical organisms, on the small side, okay, would be things like rickettsia, mycoplasma, and chlamydia. And then, kind of the mid-range things that would, what I like to call the garden variety type bacteria, E. coli, Staphylococcus aureus, things like that. We have in the mid-range. Then we have, the very large organisms. So we have, Pulophysium, very large. We have Thiamargarita, also very large. Each one, we have one is gram-positive, one is gram-negative, and they can approach the sizes of the 750 microgram more. And so when they were first identified, they were thought to be eukaryotes, but it turns out that they're bacteria. And so, in other things that we look for characterization, the single chromosome, etc., the environment to find things in, the dependence, depending on where they are, pH, salt concentrations, things like this, we can use to differentiate organisms. We have the extremophils, okay, the extremophils, these organisms that live in very, very harsh conditions, whether it's pH or temperature, osmotic pressure, things like that. One of the most extreme organisms is Deinococcus radiodurans. And again, gram-positive has 2 chromosomes, has a huge plasmid in there, okay, with a smaller one, and it's a polyextremophile. Right, so it can withstand, radiations that can kill large numbers of people. And so typically when organisms are subjected to radiation, we will have damage and breaks to the DNA, right? So in this organism, having the 2 chromosomes, if damage, occurs on the one, right, it can be fixed while the second one is still allowing for expression of proteins and synthesis of things. So it can withstand, this kind of damage. And so it's a very extreme organism. And then other things that we use, to kind of differentiate and characterize bacteria, the energy source, that the cells are using, and whether, of course, they're aerobic or anaerobic, and, and amongst those two, if they're obligate anaerobes or obligate aerobes, or they're facultative, and, if they're micro, aerophilic, on the scale of the requirements for oxygen, we can use those things to do that. And so we want to kind of see some of the other things that can be done with the cells. So gram staining we already mentioned. Okay, we'll do the gram stain. We can tell if we have gram-positive or negative. And this is based on the cell wall. So the peptidoglycan, okay, on the cell wall is what we use, okay, really in the gram stain. So who remembers how a gram stain is done? Anyone remember how a gram stain is done? Somehow, somewhere? How do we do a gram stain? Anyone remember? So okay, so I have a plate over here of bacteria growing. I want to do a gram stain. What's the first thing I'm going to do? Okay, so I'm going to make a smear, right? I'm going to let it dry. I'm going to heat fix it. Okay, and then what are the dyes I need? A crystal violet, safranin, Okay, so, well, one more thing. Okay, we need that, and then so we need this, and then we use the alcohol, what we need. So we have the alcohol, we have crystal violet, we have saffron in, we've already made our smear, we've already fixed it, right? We need one more thing. One more thing.
Grams iodine.
I heard it.
Grams iodine.
Grams iodine, right? Right. So we do the smear, we apply the crystal violet, right? Right, we stain, wash off, add the iodine, we have the mordant, right, to hold the dye in, right? We do the decolorization step, we put the saffron in, right? And so at the end of the staining process, if the cells are stained red or pink, what's that? Is that gram positive or gram negative? Gram negative. So if they're stained bluish purple, then they're gram positive, right? Okay. Excuse me. So gram staining is one of the most important stains you're going to have, to work with bacteria. Okay? So every clinical lab, okay, is going to do a gram stain. Okay? And it doesn't matter, and if so we can extend it all the way to the food industry, and everybody that has to do anything with bacteria is going to do a gram stain. Okay? So it's very important for diagnosis. The gram variable organisms, right, we can do the acid fast. So the acid fast, uses the carbonyl fusion. We have to heat to get the dye into the cell because of the waxy components we have in the cell wall, okay? And so staining is very important, okay? Two major staining techniques, gram staining, Zeal-Nielsen acid fast, and then the antigenic properties that we have, on the bacteria in the cell wall, for gram-negative cells, that we have, in LPS, and so on. All of these different things can be used to type the cells. So we can identify strains, okay, of E. coli, or strains of Staphylococcus aureus and things of that nature. So we already said, proteobacteria are pretty common, okay, amongst the, the five very diverse. But then we have the proteobacteria, we have the Bacteroides, we have the Firmicutes, Actinobacteria. 4 major phyla. So we want to make sure we're remembering them. We have some other phyla that are represented. We mentioned the TM7, okay, which, we can't culture, and so on. We have cyanobacteria, fusobacteria, etc. So we have other phyla that are there, but the 4 major ones, we want to make sure that we're remembering these right here, okay? And then, we already mentioned the fact that even for those that we cannot culture, we're using the 16S, ribosomal RNA, okay, for PCR, we identified, and we can sequence, and we can blast the sequence information, and we can find out, what kind of organisms that we have there. And so that's a very important thing that happens. So again, this kind of shows you how that would work out so that you can design the primers, for the RNA sequence, if you can use them in your PCR, okay? And so again, this basically just shows you, the ribosomal RNA, from which the information for the primers is obtained. And so if you have the DNA, right, and you have the primers, you can do the PCR, you can, sequence, the product, and, you can put it, in the databases, do a blast search, and you can identify, matches into the database, okay? And so typically when you get the readout, after you do the blast search and the readout shows up, it will show you, at the top of your list, the ones that had the highest hit, okay, all the way down to the ones that may be just spurious, at the very end. So this can give you an idea, okay, as you look down at your scores, as you look down at what's listed from the top coming down, you can see the closest matches, okay? And so, right, so again, this kind of just shows you pretty much the same thing. We're not going to belabor the point. I think we get the idea, of being able to get this information. So again, the important things to mention over here is that if we don't have the cultures, we have culture-independent ways of still identifying the organisms, right? Does that make sense? We can identify that way. And so we can, get this, metagenomic information, right, from the DNA and construct libraries. And so these libraries can be screened. Okay, so if you find something new, you can screen it against that and see if it's already been identified before or not. Okay, so it kind of makes things a little easier. And so there's a term we all want to know. Okay, and so we're looking, we already have the diversity, the number of species and everything, and we're trying to apply it, to identifying bacteria. We have some challenges with that, okay, because again, the time for cells to divide, we're having transfer of genetic information. All of this can affect what we're trying to do with the bacteria. And so this term, operational taxonomic unit, okay, we need to know. And so this is, represents, group of organisms that shares at least 97%, of the small subunit RNA sequences, okay? And this is, again, what's being used for the gene sequence identity. And so, it is not like the perfect situation, okay? But, pretty much that's kind of what we have. And so the idea is that, there's a need for better ways to even do the identification. Does that make sense? So we have stuff we're using, we can get information, but it's not like a perfect situation, so there's still room to improve on these techniques. And as we're going to find out, in the oral cavity, that's a big issue, because we have organisms in there for which we don't know, we can't say, okay, they're shaped like this, or they grow like this, or whatever, but we have the DNA information. And so if we have situations where that organism might be important, and if you're coming after it, okay, trying to, get rid of it with whatever treatment you're using, right, you're kind of doing it, like a trial and error, okay, because you're not really sure what they look like, okay? So that's why more information is needed. Now, I only have this here just to make a point from the paper by Vermeer and group. And so what they've been looking at are the regions on the 16S ribosomal RNA, okay, to find areas that are highly variable, areas that are kind of conserved, right? Since this is what's being used, to do the identification, right? Are we choosing those areas that would best give us, the right information? And so this basically just shows you that when you look at it, you have a little variability in the groups. And so, even though we're using it, we can get the information. It's still important to know that we have this, extent of the variability in the structure. And this just kind of shows the profile of how things are done. And so I'm just going to let you guys look at it, later on. Just showing you everything from identifying, the communities you're going to sample, all the way to getting the DNA information, and then doing, the blast searches and doing the analysis. So just kind of take a look at it. We'll talk about it again. But, I'm just going to wait for you guys to look at that. All right, so let's look at the differences in the gram-negative, gram-positive situation. So these are the things that we want to kind of remember. There's a lot that we can talk about, but we want to make sure that the basic criteria for having organisms, gram-positive, gram-negative, that we remember those things. And typically those are the kind of questions that, get asked, to make sure that those fundamental things are there. So with the gram-positive cells, right, we just talked about how to do the gram stain. Everybody kind of remembered that. So if we have, the cells at the end of the gram staining procedure, if they're bluish purple, we said that those cells are gram-positive. If they're reddish pink, they're gram-negative, right? So we have, in the gram-positive cells, we have, the peptidoglycan layer is very thick, okay? And, the other name for peptidoglycan is murine. So when we said the archaea have a pseudomurine, they don't have like the typical peptidoglycan, structure in the cell wall. And so in that thick cell wall layer, we have glycans We have, glucanac, so, we have the N-acetylglucosamine. Okay, we have the Munac, also as part of that is N-acetyluramic acid. We also have teichoic acids as part of the gram-positive cell wall. And we also have lipoteichoic acid as part of that cell wall as well. So these things, kind of distinguish the gram-positive cell wall from that of the gram-negative. So remember what we mentioned, right? So after you put the crystal violet, and then you wash and you add the iodine acts as a mordant, okay, M-O-R-D-A-N-T. And when you do staining procedures and you have a mordant, the mordant helps to hold the dye into the cell. And that's what's going on. So that when you come back and you decolorize, right, and kind of wash that off, anything that's not held, will wash away. And so when you stain with the safranin, those areas that are not stained take off the red dye. Okay? And this is solely the cell wall component that's allowing us to do this. Okay? If you look at the gram-negative cells, the peptidoglycan layer is very thin. And we have LPS, or lipopolysaccharide, in that cell wall. We also have periplasmic space. We have an outer membrane. We have, a lot of, channels and pores in the membrane, very different from the gram-positive cell wall makeup. Now, depending on what you're reading, these days, you will see discussion about periplasmic space in the gram-positive cells, typically small, okay, but it's a prominent feature of the gram-negative cells. And so when we think about gram-negative cells, we're thinking of things like E. coli, right, Salmonella, Shigella. Everybody with me? A whole bunch of the enterobacteria are gram-negative organisms. And so they would have this, kind of an organization. And so what you would need to remember is, so if you have a question, later on asking you have a list of the features, can you identify if they belong to gram-negative organisms or gram-positive organisms, right? And we'll look at how the cell wall, is put together, okay? And we'll see how the peptidoglycan is built. But in any case, we have the gram-variable organisms, or the ones that don't really stain. And so again, we have the cells that fall in that category over here. The archaea fall in that group. The mycobacteria, so this is the Mycobacterium tuberculosis, leprae, right? The acid-fast causative organisms. And mycoplasma, those people have a cell wall. So these are going to be gram-variable or will not stain. Okay? But, the ones with, competent, organized cell walls of both gram-positive, gram-negative, we can distinguish them based on the staining. Now, if you were staining and you decolorized way too much, right, you could wash off the crystal violet as well. And if the cells stain, pink at that point, they're not truly gram-negative. It's just the, the step, of the decolorization that was just extended for too long. So we need to know those. So again, we're looking here gram-positive, gram-negative, and I think we can appreciate, okay, when we look at the two, so we're looking, at the peptidoglycan layer, pretty thick, okay, and then we have, within the gram-negative cells, again, very thin, and so we can see, as we look inside, things like, the DNA, ribosomes, all of those things are pretty common, to both cell types, right? So the places that we're looking for those differences is the peptidoglycan, the periplasmic space, the outer membrane, to tell us that we're looking at gram-negative cells and not gram-positive, okay? So this is, a good picture, sure there are better ones, but, to help you just kind of look at a glance to see the differences, okay? And we will look at the cell wall in a little bit more detail. And the reason why we do that is because, again, many of the antibiotics that are used for bacteria are aimed at the cell wall. So if they disrupt peptidoglycan synthesis, right, they can, kill the cells that way. Okay, so that's a very important feature of bacteria that we need to have an understanding for. So again, seeing the two, right, we can see the differences that we have in there. Other things that we can find, in common, both cell types, gram-positive, gram-negative can have a capsule. Capsules are important, for immune response. because the bacteria can evade, the immunity by having that capsule over there. It makes it difficult for phagocytosis. So those we can have, in both. We can find inclusion bodies in both, gram positive, gram negative, and so on. So you have some things that are found in both, but you have things that are pretty distinct. And the cell wall is one, right? And the outer membrane, periplasmic space, etc. We're looking at the gram-negative cells. And so we all know the gram staining now. So if we see, the pink red, we can tell positive, negative. No problem with that. Okay. And again, this is just kind of reminding us of what we've been talking about. We're not going to dwell on it. And so again, as much as we have bacteria present, in the mucosa, skin, okay, different parts of the body, right? We have, a few areas where we would not expect, to see, tons of bacteria. Okay, so within the tissues, when we're looking, we have areas that, essentially we could regard as sterile. And then, of course, the mucosal areas, depending on where we are, we have bacteria in those locations. And so this is just to kind of remind us of that. We have the tables over here for today. They just, the tables are just tables to us today. We just have a bunch of bacteria listed on there. We don't have to worry about them beyond that. But we're going to begin to narrow things down, okay, to where we're going to say, okay, these group of organisms, definitely we know why they're there. We know how the biofilms form. We know who gets there first and, who gets recruited and so on. So for now, we're just looking at, a bunch of organisms. So if we look at the gram-positive cells, we have examples of them. Everything from Streptococcus, Staphylococcus, Lactobacillus, Bacillus. These are all gram-positive, Clostridium. So we'll begin to recognize them, okay? And we'll also begin to recognize them in terms of whether they're aerobes or anaerobes. And then amongst the gram-negative, we already mentioned, the Enterobacter. But then, we're going to see, we have, the Porphyromonas. We're going to talk about Porphyromonas a lot. And then, so on. So these are all gram-negative organisms here that we'll see. Amongst, the distribution, we have the resident microflora. We have those that are transient. So which means a lot of the investigations being done to understand how we get colonized are being done in geographic areas of the world. Right? Why would that be important? Why would you want to know the kind of distribution we have for the microbiome in different geographic parts of the world? Why would that be important?
To understand the spread of disease and how that relates to the menstrual bacteria.
Okay, that's one. Okay, that's one. What else? Okay, so understanding disease distribution is, is important because you want to see if you're seeing the same organisms. So a person comes in, they have an infection, we have inflammation going on in the mouth, whatever. Is it the same organisms that are causing this, or do we have different things, right? If it's the same, then are we looking at the same variants, the same strains, or do we have geographic strains, right? But what else? This is a huge area of research, trying to understand how we get colonized from birth. Okay, anything else come to mind?
The immunity of people in those geographic locations to certain.
So that's related to the disease distribution, right? So think about it. So the question is, do we have core organisms that regardless of where you live geographically, they're going to be the same? Okay? Or do we have, vast differences in the core makeup, right? Because we talk about organisms, that are normally present, and then we talk about transient organisms. So if you travel today, to Lagos, and in Nigeria, and you're there for a couple of days, you're going to have transient bacteria that are going to show up from that environment. Okay? They're not the ones that you find, here normally, but you're in a new location right now, right? If you travel to Glasgow, Scotland, a couple of days, you're going to have some changes, right? So there's core, and then there's transient, okay? And so if things are transient, unless they're a problem, right, it's like, okay, we know that's gonna happen, but it's not a big issue. But if you're going to have, a transient situation where you're going to have opportunistic organisms that can actually cause a problem, then yes, we want to know about them, okay? And so we have colonization, okay, right after birth. We have different organisms that colonize the child, whether the child was born vaginally or whether they were born by cesarean section. So you're going to have those differences. And then as the child grows and develops within the immediate environment, they're going to have organisms that essentially look like what everybody else in the family, okay, has. And some of these are going to be different, from other individuals, right? But then they're going to be those core organisms that we expect, should be there in that location. So the area where that allows for this to happen, the mucous environment, we're close to the outside. So we're going to have this transient organisms, bless you. And then individuals who are immunocompromised, we're going to have some differences there as well. And then we're going to see medication, depending on what a person is taking, we can have alterations. And if we do damage to the relationship, that we have, we're going to have a dysbiosis. That's another term that we want to kind of make note of. And if we have that dysbiosis, right, we can have a pathologic situation, okay? And this is true in the oral cavity, it's true in the GI tract, it's true in the urogenital tract, and so on. So we want to kind of remember that term, dysbiosis. And so if we look at these organisms that we have living in and on us, okay, and if we kind of compare that, we have symbiotic situations. We don't have true endosymbiotic type of things, but we can have organisms colonize body cavities and so on. The skin is really good for that. GI tract is really good for that. And so outside of the molecular stuff that we just talked about, another way that these organisms are studied is using nonabiotic animals. So these are germ-free animals. So you can actually populate them with the organisms that you want, and you can study the interaction of those organisms in those animals. And so that's done to kind of get a better understanding of how the different organisms interact with each other, okay, and to be able to get a profile out of that. So again, we have the common areas. We want to make sure we remember them. So if you see a list of the places, you want to be able to identify that organisms can be found on the skin, the digestive tract, oral tract, urogenital tract, etc. Okay, and we can find the organisms in those locations. And so, on the skin, we can have them on the surface, we can have them in the hair follicles, etc. And again, when these organisms are looked at, the same thing we've been saying over and over. So if we, forget everything else today, we need to remember the four phyla. Okay, so we want to remember those four phyla. And so again, we're seeing that again we have this representation of these four. And so of the group, we have the two gram-positive ones, right? The actinobacteria, the firmicutes, and then we have the large, proteobacteria group, and then the bacteroides. So we need to remember these four phyla. We need to remember, whether they're gram-positive or gram-negative. Okay. Okay. So, if we look at a few of these places just to see what it is the organisms are doing. And so before we do that, we can take like a 5-minute break or something. If you want to stand up and stretch, get a drink of water or something, and then we'll come back and we'll continue. And so we have also moisture levels, hormonal influence, that have to do with the age of the individual and so on. And so Staphylococcus species, are found, and epididymis is one that's pretty common. We can also find E. coli and Enterococcus, Candida, which is a yeast organism, fungi, also in this area. So again, the proximity to the environment, and the changes, in the pH, kind of account for this. And so one of the major roles that these commensals have for the body is that they can actually help to prevent pathogenic organisms from taking hold. So they can do this physically, just, by the space, or they can actually synthesize substances that will retard the growth of, these pathogenic organisms. And so if there are situations, where, for example, say pH is altered by a pathogen, right, then the commensals, really have a hard time helping, especially if the numbers of the pathogen are higher than, the commensals in that location. So that's an important thing to keep in mind. And this is true in the GI tract, it's true on the skin, in your genital areas, etc. Wherever you find these organisms, they have a use like that. In the oral cavity, so if we looked at the two locations in the body that have the highest number of bacteria, right? We have the GI tract, and then we have the oral cavity. These two places, okay, have the most. And so we have, the environment that's conducive for this, warm, moist, nutrient-rich. We can find, strict anaerobes, facultative anaerobes, we can find some aerobic organisms. And over 700 bacterial species detected in the mouth. So that's a lot of organisms and a lot of DNA, okay, for the metagenomics that we mentioned. And a lot of this, again, is using the 16S ribosomal RNA gene, okay, to detect these organisms. So even, the ones that we cannot grow, okay, so we can detect them. And so we have both gram-positive and gram-negative organisms represented in the oral cavity. So we have, Streptococcus mutans. You'll see us mention this over and over again. We have, Fusobacterium species that will detect, below the gum line area. And then we have Porphyromonas gingivalis. Like I said, we're going to be mentioning this over and over again. There's a lot of, studies that are being done, to look at this particular organism and other bacteria that are found associated with it. So, so we have, the example shows over here, in flat deposits, we have organisms that we always see. And so there's an interesting area of dentistry, dental research, where you have people investigating, to find out, if our ancestors had the same profile of bacteria that we have today. So they're examining, teeth, obtained, from archaeological sites. They're looking at the, food deposits that are still in there that can still be recognized, and they're amplifying DNA from these specimens and finding that some of the same organisms we're concerned about today that we're talking about were present even then. And, and the differences, that are being found have to do with changes in the food types, okay, that people started eating, okay, changing the profile of the bacteria that were now, being identified. So again, very interesting area of research. It shows, how we can have colonization. On here, we'll spend a lot of time looking at this over here, but pretty much it shows you that we have the organisms, that initiate, the colonization, and then you have successive organisms, that now will show up. And so if you look at the far end over there, you see that your Streptococcus species, usually kind of show up first, and then we have the additional organisms that now come in, and then on the peripheries of the biofilms that are formed, you're going to find those transient organisms, that may or may not be there, depending on, on who you look at. So we'll see how this happens to form the biofilm. And so a little bit closer looking, just kind of showing what happens, as we get biofilm built up inside the oral cavity, right? So we can see, the way the organisms kind of organize themselves, right? The more, more anaerobic organisms are found deeper, okay, within the biofilm and work their way up. And so we can see, differences between the early part of colonization and then later in the event where you now have a more mature biofilm, right? And then you can see that the products, from those cells contributes, to the establishment of the biofilm. And one of the organisms that's most important in this is Porphyromonas gingivalis. So this is one that we want to kind of keep an eye on. Okay, we're going to need to know stuff about that. And so when we find Porphyromonas, we also ask, what are the other organisms that are associated with it? And one of the ones, a species of Piperina, can be found with the Porphyromonas. So one of the Porphyromonas gingivalis. And so we can see other things as, we get the plaque buildup and so on. So this is a major problem, with caries and things like that, and we'll talk about that some more. But pretty much, it's just showing you, how much damage can begin to ensue once you have these organisms present in those locations. So, both, caries, various aspects of periodontal disease, all bacteria related. If we have inflammatory responses that are ongoing as the immune system tries to respond to the bacteria and bacterial products, then we can have cell recruitment, and then we can have additional damage that will occur. And we will see that when we look at the different types of disease. Within the GI tract, again, very large numbers, okay, between 800 to 2,500 of bacteria, plus the bacteriophages, okay, we should not forget that. And then, other things that will lead to the change in the behavior, right, the diet, all of those, can affect the populations that we have within the GI. And so in here, what you want to do as you're looking at the organisms, right, so as you have them listed, you want to see the different types, that we have. So again, the 4 phyla that we've been trying to emphasize for today, we see that we have them there, okay? And then so, amongst the different genera and so on, you see that we have a very broad representation within the GI. Okay, and so again, this table, just kind of summarizes some things. Again, just take a look at it. And the key thing, 2 things from this table that we want to kind of pay attention to, right? What the gram reaction is for these major organisms, and what it is that they're doing, okay, within that GI. Okay, so you want to just kind of take a look at that. And other things, that questions that come up, and this is true wherever we find the organisms, right? Do we find them just external to the tissues? So if you look at the epithelium, okay, in the oral cavity, we look at the epithelium, the GI, right? So, we have this, columnar, epithelium that we have going throughout, the whole GI tract. We have the stratified squamous, epithelium we have in the oral cavity. In some areas we have, keratinized, right? So with this distribution, what's going on with the way the bacteria finding themselves? Are they attaching? Are they trying to do anything beyond that? And, why is it that, again, when we begin to have these biofilms form, in some areas, they're very tough to remove, okay, once they form. So when plaque forms, it's pretty tough, but you can find the organisms mixed in there. So if you were to take that sample and if you were to either, try to grow it or get DNA from it, you can get, enough information just from that. So, when we look at that, the fact that we can wash some of it off, as tough as the biofilm may be, okay, those situations where the organisms are pathogenic or they're invasive and they're trying to actually get into the tissues, right, those would be the situations where we have, any kind of attachments going on. When they form the biofilm, they produce substances to help them hold on, okay? And so, we'll see examples of those. And we'll see this, not just in the mouth, but GI tract, genital tract, we'll see these communities of organisms, that will be in those locations. We have, things that are beneficial, right? So organisms, produce fatty acids, metabolites. We can have vitamins, that we absorb from them. They can assist in digestive products, so we have a lot of things that normal commensals would do. And so we have, again, when we have those changes that occur, when we don't have certain populations of the cells, in the GI, right, we can have other problems resolved. So the hygiene hypothesis, anybody heard of that? Everybody's aware of the hygiene hypothesis, right? So that's, an area of, great interest because some of the allergies and autoimmune diseases, that we're familiar with, around areas like this, in the U.S., in areas where individuals are exposed to helminths very early, okay, and they get worm infections and things like this, and the immune system deals with those allergens and all of that. There are certain things that are not seen as frequently, and asthmas, allergies, and a lot of autoimmune conditions. And so a lot of the conclusion, is that, these changes in the populations of the cells, okay, is, responsible for that. To the point where, you have exposure to non-pathogenic helminths, okay, that are being used therapeutically to have the immune system, respond to those things, to help prevent, the crises that can occur when individuals go into asthmatic reactions and things like this. So that's a very important area of research as well. Okay, so we have, again, for right now, this is just a list to you, okay, and we're going to pull things out. And so when we look at the microbiome, we have a whole bunch of different organisms. Now I have the Giardia in red because in some of the publications you'll see them mention Giardia, but Giardia is not a true commensal organism in the GI tract. So people can, get Giardia infections, and it's symptomatic. The Giardia will be present there, and they will not have diarrhea. They will not show any symptoms, but the Giardia will end cyst. So which means they can contaminate places with the cysts. If they're working with food, they can contaminate, the food with cysts, from the stool, and then individuals who are susceptible, will not get very sick. So Giardia can be found in non-symptomatic individuals, and they don't have full giardiasis, but they can contaminate and kind of spread that around. Okay. Similarly, we have other organisms that are indicator organisms, also protozoan, and Endolimus is one of those. And individuals may not show any symptoms, whatever, but they can transmit to others who become more susceptible. And so it's important to mention those in there because, again, depending on where you're seeing patients and depending on what else is going with them, these organisms, will show up. One that's a little bit of a surprise is Helicobacter pylori, that, some individuals in the oral cavity, swabs and so on, it shows up, but it really should not be there. You expect it to be lower down, okay? And so it's important to know what else can be there. And so, again, right now this is a list, and we're going to pull out from here the things that we're going to need to work with. In the oral cavity, again, we're looking at a distribution. Again, we see amongst the bacteria, there are two protozoans over here that, depending on where you're practicing dentistry, you might find that the individuals have a lot of these protozoans in the mouth. And so we have Entamoeba gingivalis, okay, which is an amoeba. And then we also have Trichomonas tenax. So one is amoeba, one is flagellate. And the way they interact with the bacteria, in the mouth is not fully understood, okay? So that's something else, just to kind of keep in mind. And then, within the oral cavity, we have the oropharynx area, we have additional organisms, okay, that we need to kind of keep track of. And so again, here, we find the same, we found, in other areas of the body, can also be found here as well. So we just need to keep track. On the skin, we have Staph aureus and so on. In the genital tract, we have some organisms that we have, in the oral cavity and on the skin. And here, other organisms that can be found, in addition to the bacteria, Trichomonas vaginalis, some individuals are asymptomatic. So which means, if they're not showing symptoms, the organism is present. If you were to look at the population of organisms you have in there, you would pick that up, okay? And so this is an organism that can alter the pH of the vagina and can actually allow itself to grow in more numbers. And so if you have situations where individuals are symptomatic and they're really they're being affected by the presence of this organism, so typically they will be seeking medical help for that. And the metronidazole usually will kill these organisms once they've been prescribed that. And so we have other places we can see a fair distribution, not as heavy as the gut or the oral cavity or even the skin, but we have a mixture of organisms around the conjunctiva. We have again those 4 phyla represented, and around the ears we can also have organisms. So these are areas that we don't usually think about things being there, but we have a wide distribution throughout. So we're going to be talking about bacteria and their types and disease-causing organisms, as opposed to the commensal ones. There are some things that we need to know about pathogenicity. So we have these terms over here. So if we can, grow the cells, typically we can count the cells. We can do all different types of enumeration type of methods, to know what's there. And so microbial count is important. The virulence of the organism is also important. So not all strains of a species, okay, may be virulent. Does that make sense? Okay, some, may not cause any, infection, whereas, if you have virulent markers and molecules, then, we can have infections take place. Attenuation, is another term we want to keep in mind. So, where we can have the organism, growing, multiplying, but no disease-causing aspect of that. And so, we know this from, older vaccine studies, and even some of the newer ones, we can molecularly attenuate organisms and have them not, continue to proliferate. And then virulence transposition, okay? So we can have, if you remember, whenever you have the bacteria, wherever you have them, they have their bacteriophage, they have their plasmids. So we can exchange, plasmids and exchange, phage and so on. So if you have the genes encoding the virulence molecules within the plasmid, and that gets, transferred to an organism that normally would not be pathogenic, we can have expression of the virulence molecule, and we can have, the infection take place. Okay? So that's an important thing. So it's one thing to know the bacteria that are there. It's another thing to know, right, that they have bacteriophage, they have plasmids, and that, in the same way that we can have virulence, transferred to non-pathogenic organisms, we can have resistance to antibacterials also transferred, okay, within these communities. And so we have, when we talk about pathogenicity, we always need to keep in mind these Koch's postulates. Okay, so everybody's familiar with these postulates? You've heard them before, somehow, somewhere. We're all familiar with them, right? So when you're working, again, we can use them. They were originally, for bacteria, but then, we find that we can use them for protists, we can use them for viruses and so on. And depending on what the organism is, or what particular kind of virus you're looking at some point things kind of break down a little bit, and we'll explain that. So the idea is, if you have suspected organism, okay, causing an infection, you want to be able to isolate it, right? You want to be able to, have that thing inoculated into clean animals and basically, see the same infection. And you need to be able to get the organism back out of that, host and culture, okay? And basically show that you have the same organism, okay? Now, this works if you have, you know, a good host where you can have the organism grow. And if within that host you can have the same kind of infection show up, okay? So if you don't have a good host, right, then this kind of breaks down a little bit, okay? Because you can't demonstrate, you know, that the infection is going to take hold and that you can isolate the organism back out, right? So you do have a few cases where that happens, but generally, okay, if you have a good host, if you're able to get that pathogen into the organism, you can satisfy, you know, the postulates. And so in infectious disease, when you're dealing with, you know, with infectious agents, you know, whether it's bacteria, whether it's, you know, parasites, you know, whether it's viruses and so on, you know, this template, okay, is what's used, okay, to kind of satisfy the fact that the etiological agent, okay, you know, the one causing the infection is really what it is, okay? So, so we want to remember these postulates, and we want to remember what they're used for. So we'll stop here, and I just want us to finish by looking over here at some of the things that we talked about. So let's see if it will work. I tried to do it before, and I think it did work. Let's see. Okay, so. And the things that we've talked about today, okay, with the bacteria. All right, so we know, okay, we're looking at prokaryotes. And so we said, you know, we can have the peptidoglycan, okay, so urine is also peptidoglycan, okay, or we can have those that have like the pseudomurine, okay. So this group over here would be the archaea. So at this point over here with the bacteria and the archaea, we're looking at the domains, okay? What's the other domain? You have the eukaryote, right? Everybody remembers that? Okay. So amongst the bacteria, right, so we have gram-positive, we have gram-negative, right? So we have that. And so we mentioned that with the gram-positive ones, we can have either low GC, okay, or high G plus C, right? And so, you know, and when we get into, you know, like next, I think we like lecture 3 or 4, okay, we're going to talk about like just really briefly, okay, you know, how we differentiate, you know, between something like strep and staph. Okay, so we're looking, and then even amongst the staff, we do something like Staphylococcus aureus and Staphylococcus epidermidis, for example, right? How we can differentiate those things. And so amongst the gram-negative organisms, right? We mentioned, so we have the, you know, the phyla that we talked about. So the 2 gram-negative ones, right? You remember the 2 phyla of the gram-negative? So we had the Bacteroides, right? And Proteobacteria, right? So we have, you know, and then the Proteobacteria.
Are you closing off just a little bit?
Yeah, open it a little bit.
Yeah.
Okay, can we see? Okay, and then among the Proteobacteria, we said we have 5 subgroups, right? Do you remember what they are? We have alpha Proteobacteria, beta Proteobacteria, delta, epsilon, gamma, right? Within the group, okay? And so all of these, right, so among the Proteobacteria, gram-negative, right? So among the gram-negative, we also have the non-Proteobacteria, okay? Also within this gram-negative group, okay? And so when we mention something like Porphyromonas gingivalis, right? You know, Treponema, we're mentioning all these different things. Number one, what phylum do they belong? Right? What's their gram-positivity? Okay, is it negative, is it positive, right? What are they? Are they rods? You know, are they Coccobacillary? You know, are they cocci? What are they? So, you know, from now on, as we're talking about things, right, we start mentioning those things. Okay, so we're going to need to know if we're mentioning, you know, Streptococcus, you know, mutans, Okay, what is that? The gram positive, gram negative, and so on. And so in the distribution, as we begin to look at the organisms, in the oral cavity, right, where are we finding them? Okay, are they predominantly on the cheeks, are they in the gum line, below the gum line, where are they? Okay, and so when we have the biofilms in the form, right, and we see, a certain group of organisms together in one location but not in the other, okay, why is that? Okay, and so we need to kind of keep track of this, this arrangement. And so the 4 phyla, right? So we've mentioned 2 over here right now, right? We mentioned the Bacteroides, we'll put it back there. What are the other 2? So we have Firmicutes and Actinobacterium, okay? So, we want to kind of remember those things as we're kind of going along so that we know. So then, for next time, the differences between gram-positive bacteria, gram-negative bacteria, just the general things. We want to make sure that we're remembering them, okay? And then we're going to look into, like the peptidoglycan, how that's put together and all of that, cell and different features, right? And then so that we can start looking at, disease causation and so on, okay? Are there any questions? Okay. Question?
Can I see the right
Oh,
side?
shift it?
Thank you.
Yeah. Okay. Okay. So it looks like we're going to be meeting in here, and if somebody else needs it, then we'll go to the other room. So we'll go back and forth depending on what happens. Yeah. Okay. All right. And so if you have any questions at any time, so if you forward them to Ashley, she gets them to me, I will respond to you. Okay. So as the notes, are coming up and you're looking at them, you're preparing and all of that, if a question comes up, do not hesitate. Okay, just let me know. And so most times, I send it to you and the rest of the class can see the response.
I'm working with Alicia too. Alicia's notating their class.
Okay, yeah, okay, good. Okay, any questions? Okay, so we'll be back tomorrow, 3 to 5.
Thank you.
Okay, all right.